at active and bulk transport goes with section 5.4 and 5.5. Active and bulk transport are just other ways to transport substances across the cell membrane. So if we think about the ways we know to transport things across the cell membrane, so far we've learned about passive transport. And that takes no energy. And in our cells we use ATP for energy. And the passive transport mechanisms we've looked at are diffusion, osmosis, and facilitated diffusion. So diffusion, really easy. What we did our labs on substances tending to move from an area of higher concentration to an area of lower concentration. And osmosis, the diffusion of water. So these are pretty standard, pretty easy to get. I think after doing our labs, at least conceptually, we all are pretty solid in diffusion osmosis. And facilitated diffusion, objects or molecules are still moving along their concentration gradient from high to low, but in facilitated diffusion, if this is our plasma membrane, so we got our hydrophilic heads and our hydrophobic tails of our plasma membrane. And what we're going to have is a protein that is in our plasma membrane. Our plasma membrane is going to continue off this way. So we have a protein in our plasma membrane, and it's called a channel protein. And molecules that are too big or too polar, too big / polar, to diffuse through the plasma membrane, diffuse through this little opening, this little channel, this little gate that the channel protein has created. That's facilitated diffusion. It's facilitated because the molecules can't either fit or are too polar, can't go through the membrane all by themselves. They just need this little channel to go through, this channel protein. Facilitated diffusion. Again, it's passive transport. It doesn't take any energy. What we're going to look at today is something else. What we're going to look at today is active transport. And the important thing about active transport is it requires energy. ATP. It requires energy. So the first one that we're going to look at in active transport is a membrane pump. So what you're going to see in active transport is kind of the same thing. We got our plasma membrane, and I'm going to just write it here so I don't have to take time to draw all the phospholipids again. And just like before, we're going to have a protein that is in the plasma membrane, an integral protein. And what it's going to do is, in active transport, it's going to move substances against the concentration gradient. So what do I mean by that? If we have a high concentration of these molecules on the outside of the cell here, so we'll say outside is down here and inside is in here. This protein pump, this form of active transport, is going to move these chemicals, these molecules, from the inside to the outside, from an area of low concentration to an area of high concentration. So in the opposite direction that diffusion would naturally want them to go. That's why it takes energy. That's why it takes ATP. So what happens is, say this protein is going to take one of these molecules from the inside and push it out, pump it out just like a pump. The molecule would kind of bind into this little hole in the transport, and a molecule of ATP would bind onto the transporter, and the transporter would change shape and move that molecule from in here outside. So it's kind of like a Pac-Man. It opens up, and once the ATP binds onto it with the molecule moving from the inside to the outside, it changes shape. So it changes shape and pushes it out of the cell, moving it against this concentration gradient. That's why it's active transport. And there are several active transporters that are really important to your body. One of them, important active transporter, is the sodium-potassium pump. The sodium-potassium pump. Now, that is really, really, really important in your body, because sodium and potassium are very important ions for cells, for membrane potential. So for nerve impulses in your brain and in your body. And your body constantly needs to keep a high concentration of sodium out of your cell and a high concentration of potassium inside of your cell. So in your body, you have a protein pump, an active transporter, kind of looks like this. And on its job is to pump sodium, to pump sodium this way out of your cell, and potassium into your cell. And keep the concentration of potassium high inside of your cell and the concentration of sodium high outside of your cell. So what's going to happen is potassium is going to come and bind on to this protein pump, and on will stick ATP, and it's going to change shape and push the sodium into your cell. So we're going to have sodium on the outside, or potassium on the outside, and it's going to push that potassium inside of your cell, and then take any sodium that's inside of your cell and move it out. So it's going to push sodium out of your cell and potassium into your cell against their concentration. There's already a lot of potassium inside of your cell, but this pump is going to push even more into your cell. So already a lot of sodium on the outside of the cell, but this pump is going to push even more out of your cell. And it's going to take ATP to do that. For the sodium to bind on to the sodium places, ATP to trigger that protein to change shape and push the potassium into your cell, and then the sodium into your cell to bind onto the sodium-potassium pump, and the ATP to change its shape, and then the sodium to be released outside of your cell. So it is responsible for, what would you call it, membrane potential, for signal transduction, for nerve impulses to travel through the nerve cells in your body. And we call a protein like this, a protein pump, an active transporter that establishes a charge on the in and the outside of the cell, an electrogenic pump. So by moving certain ions inside and certain ions outside, it establishes a differential in charge from the inside and the outside of the cell. Another example is the H+ pump, or the proton pump. And what it does, got our membrane again, and we've got our H+ pump, is it takes hydrogen ions and moves them all in one direction against their so it's establishing a big positive charge on one side. And this is going to be really important in cellular respiration, which we're going to talk about in our next unit. The proton pump basically is responsible for your electron transport chain generating all that ATP. So in cellular respiration, this proton pump, an electrogenic pump, active transport, needs a little ATP to get working, is going to be very, very important. Sometimes active transport and passive transport can work together. Sometimes active transport and facilitated diffusion can come together in what's called a CoTransporter. And in a CoTransporter, what you have, and I'll do another picture of it, we've got our plasma membrane, and I'm going to have a protein like that, and on the inside of the cell we're going to have a bunch of hydrogen ions. And on the outside of the cell, we're going to have a lot of glucose. Let's pretend it's glucose. And the glucose has a high concentration out here and a low concentration in here. The low concentration. The protons have a low concentration in here and a high concentration out here. Basically what a CoTransporter is coupling passive transport, diffusion, facilitated diffusion, and active transport together. So what it's going to do is it's going to allow the glucose to diffuse with no energy along its concentration gradient. It's acting like a facilitating diffusing protein, a carrier channel protein, to allow the glucose to diffuse along its concentration gradient from high to low. And that's going to store up some energy inside this CoTransporter. And this CoTransporter is going to use that to simultaneously pump hydrogen ions out against their concentration gradient. So CoTransporter couples facilitated diffusion with active transport. So every time a glucose molecule diffuses through this channel protein, the channel protein is going to use that and pump a hydrogen ion out against its concentration gradient. So they really couple active transport and facilitated diffusion together. That's what a CoTransport is. Alright. The last thing that we're going to talk about today is called bulk transport. So, moving more than just a few molecules at a time, but a large amount of stuff at once. And we do that in two ways: exocytosis and endocytosis. Exocytosis moves things out of the cell, and endocytosis moves things into the cell. And this is how it's going to work. This is how the two are going to work. So here we have the plasma membrane. If we're going to move things out of the cell, exocytosis, so this is especially common in cells of organs that secrete a lot of different substances. So like the stomach cells with the stomach acid, or the pancreas with insulin. How they secrete those substances, say this is a substance, is that those substances are traveling about in vesicles. And the vesicle is going to come up right against the plasma membrane and sort of fuse with the plasma membrane, form a little pocket like this, and kind of the vesicle is going to join up, yet fuse to the plasma membrane and push the stuff out of the cell. So now this little vesicle has joined, fused to the plasma membrane, pushed the stuff out of the cell, and now we've got the stuff out of the cell, the insulin or whatever we have it, and our cell membrane stayed intact the whole time. There never had to become a hole in it. It never had a break. That vesicle just fused with the cell membrane and expanded outward and pushed the stuff out of the cell. That's exocytosis. Endocytosis. Alright, so this, I'm going to just, so we don't get confused. Oh, my bad. This up here is our exocytosis. Our endocytosis is the reverse. Endocytosis is the reverse. And one example of exocytosis is cholesterol. So, the surface of cells, like we talked about, have these receptor proteins. And cholesterol is a molecule that's outside of the cell. And when they come, and it's trying to get into the cell, when it comes and binds with these receptors, what happens is the opposite of exocytosis, the plasma membrane starts to form a little kind of convolution, little indent in it, where those cholesterol molecules are bound onto those receptors. And what will happen is that little pocket will eventually pinch off and become a vesicle. So those cholesterol molecules bind with the receptors, the plasma membrane starts to bend in, form a little pocket, and then the pocket's going to pinch off and form a vesicle in the cell, and now that cholesterol is in the cell. That's endocytosis. And since these cholesterol molecules are binding to these receptors, and that's what's telling the cell to start creating this pocket to do some endocytosis, when there's these receptors, it's called receptor mediated endocytosis. Receptor mediated because these molecules wanting to get in have to signal these receptors, tell them, all right, we're going to do endocytosis to pinch off the plasma membrane and bring them inside. Sometimes that doesn't have to happen, so that there's not receptor mediated, but this example is one of receptor mediated endocytosis, bringing in molecules that way. So, what we learned today: exocytosis and endocytosis, two forms of bulk transport to get things in and out of the cell. We learned about CoTransporters, kind of coupling active transport, moving things against their concentration gradient from low concentration to high concentration, coupling that with facilitated diffusion. So every time something diffuses naturally across its concentration gradient using a CoTransporter, the CoTransporter pumps something against its concentration gradient using active transport. And then we talked about active transport. The sodium and potassium pump, that is an example of active transport, moving ions against their concentration gradient, needing ATP energy, and how it's an example of an electrogenic pump, like the H+ pump, that establishes, by moving things against their concentration gradient, a charge on one side, inside or outside of the cell. Alright, that is it for active and bulk transport 5.4 and 5.5. If you have any questions, let me know.